Now that we've covered some basic background on amino acids, let's start connecting them together with the peptide bond. We're going to start with some basic vocabulary, so when these words come up, you know what they mean. So a polypeptide is a single chain of amino acids that are linked by peptide bonds. The peptide bond is the bond between the amino acids. The peptide bond is also called an amide bond, so if you see that, it means the same thing. Every amino acid in a protein is called a residue. They're not called residues when they're free amino acids, but once they're linked into a protein, they can be referred to as residues. And then what's the difference between a peptide and a protein? There isn't a very big difference, except that a peptide is short and a protein is long. So a sort of an arbitrary definition I found is that a peptide is less than 50 amino acids, while a protein is longer. Let's take a look at that peptide bond. So the amino acids are covalently linked by that peptide bond, where the carboxyl from one amino acid interacts with the amine from another amino acid. We're going to have water as a product of this reaction. And here is our newly formed peptide bond. So when we're looking for this, we're going to look for that carbonyl and the amine, and the bond between them, that's the peptide bond. Notice in our newly formed peptide made out of two amino acids, we have what's called the N-terminus. So that's going to be the end of the peptide where the amine is. And we also have the C-terminus, which is where that carboxylic acid is. So let's take that and apply it. So as our peptide chains get longer and longer, we're going to start adding more and more and more and more amino acids. So looking between these, we should be able to identify the peptide bond connecting each of these amino acids. So if I look for the peptide bond connecting amino acid 1 with amino acid 2, I can see there it is between the carbonyl and the amine. If I look for the peptide bond connecting amino acid 2 with amino acid 3, there it is between the carbonyl and the amine. So we're going to see several peptide bonds in this peptide, and that's important that you can identify where the peptide bond is. We could also identify the alpha carbon. So here is the alpha carbon for amino acid 1, here's the alpha carbon for amino acid 2, and so on. So reading a peptide sequence, here is another peptide sequence. We've got 5 amino acids. We are always going to read these from the N-terminus to the C-terminus. So the N-terminus is also called the amino terminus. You might hear either of these words. So our first amino acid, we've got our alpha carbon, we've got our side chain here, our R group for tyrosine, and then we've got our peptide bond connecting amino acid 1 to amino acid 2, which is a glycine. Here's our R group, just a hydrogen. Here's our next peptide bond, etc. So as we go through, it's an important skill that when you see a peptide, you can identify the N-terminus of it, the C-terminus, or the carboxyl terminus of that peptide. You can identify the R groups and the alpha carbons and the peptide bonds. So peptides are always read from N-terminus to C-terminus. That matches up with the order in which they were translated. So whenever you're given a peptide, the N-terminus is always the first amino acid in that peptide sequence. I also want to point out, notice that these N-terminus, the N-terminus and the C-terminus are always charged. So while the peptide bond is uncharged, the termini, N and C termini, will always be charged. This is the protein's primary structure. So when we talk about protein folding, we talk about primary, secondary, tertiary, and quaternary structure. The protein's primary structure is just the sequence of amino acids that are covalently bound to each other. No other folding, no alpha helices or beta sheets, nothing. It's just the sequence of amino acids covalently bound to each other. So when we talk about the primary sequence, it just means we have tyrosine, glycine, glycine, phenylalanine, leucine bound together, for example. All right, now we're going to take a closer look at the peptide bond and some of the movement it can and can't have. So a quick refresher from some basic chemistry. Remember that if you have a single bond, say between these two carbon atoms, that those groups can rotate freely around the single bond. So if we're tracking, for example, this hydrogen, we see it can start at the top, rotate down to the bottom, rotate to the back. Those single bonds rotate freely. They do have certain preferred geometries, but they can rotate. Now that's in contrast to double bonds. Remember that double bonds do not rotate freely. They have two different isomers that they can adopt, but that they're not rotating between those. So remember the trans isomer is where larger groups are going to be on opposite sides, whereas the cis isomer means that the larger groups will be on the same side. So when we think about the peptide bond, I want to be clear, this is the dominant structure of the peptide bond. So it is a single bond. There are no charges. But what makes this a little bit complicated is that the peptide bond undergoes some resonance with that carbonyl right there, where that those electrons from that bond can pop down here and be shared between the carbon and the nitrogen. So because the peptide bond has this resonance between these two different structures, it means that it actually has some partial double bond character. And because of that, the peptide bond does not rotate. So all of your peptide bonds will be in either a cis or trans conformation. So let's take a look at this one to start. Is this peptide bond in cis or is it in trans? So let's look for our bigger substituents here. So here's our bigger substituent, and on the other side, here's our bigger substituent. These are on opposite sides, so this peptide bond is in trans. Just to show that a different way, again, here's your peptide bond. When we have the trans conformation, like what you see on the left, your neighboring C alpha carbons are on opposite sides of the peptide bond. In pink here, that's your peptide bond. When you're in trans, these are pointing in different directions. Versus when we have the cis configuration, now our alpha carbons are on the same side. So this peptide bond is in cis. While we're here, I want to also introduce you to the coloring scheme that's used for these images that you see down here. So this is called CPK coloring, and it's a really commonly used coloring scheme for drawing molecules. Maybe you've seen it before, maybe you haven't. So if you haven't, here's a refresher. The gray balls, those are going to be your carbon atoms. Blue will be nitrogen. Red, or kind of pinkish in this diagram, are oxygen. And then white's going to be your hydrogen. So what that means is if you see a diagram like this, you can interpret what each of these atoms are based on their CPK coloring. Another interesting feature of the peptide bond is it's planar. So 6 atoms in the peptide bond and surrounding it lie in a plane. So that includes your alpha carbon from one amino acid, the carbon and nitrogen in the peptide bond, the oxygen and the carbonyl, the hydrogen and the amine, and then the next alpha carbon. So all of those atoms lie in a plane, which makes for some interesting geometrical constraints. And then just to practice, is this peptide bond trans or cis? This peptide bond is in trans because here's our alpha carbon. They're on opposite sides of the peptide bond. All right, so here's a practice problem. You can work to review that information. Draw 2 amino acids connected by a peptide bond in the trans conformation. For this problem, you can use R, just the generic side chain, to indicate the side chain. Make sure you draw all atoms present, including their charges. After you've drawn your molecule, label the peptide bond, the amino terminus, and the carboxyl terminus.